It's a real pleasure to introduce Morten Allentoft to talk about ancient DNA preservation. I've read your papers for a long time, never met you in person, so welcome.
Thank you. Is this one on? Or? Okay, here we go. Thanks very much for inviting me to talk here today a little bit about our research that we're doing at Center for Geogenetics at the Natural History Museum of Copenhagen in Denmark. So I'm an evolutionary biologist. I don't know a lot about biobanks and storage facilities and these types of questions, but I'm mostly interested in evolution from a molecular perspective and using ancient DNA to understand that. In particular, these past few years, I've worked a lot on ancient humans, how humans have migrated across the globe and colonized the various areas and regions, and using ancient DNA to understand that. But of course, as a byproduct in a lot of the research we're doing, we get quite a deep understanding of how these biomolecules degrade and decay over time, because we really need to understand that process in order to sample the right way and in order to assess the collections in the most profitable way for all the downstream work and analysis. And this is what I'll be talking about today, some of the insights we have gained in recent years on ancient DNA preservation and what factors that are involved. So first of all, the first question you can ask is, of course, what is DNA preservation? And there are many aspects to touch on under that title, but you can say there are certainly two important aspects. The first one is fragmentation, DNA fragment length, because DNA fragments over time. It's an unstable molecule. It's getting constantly attacked by various spontaneous hydrolytic oxidative processes and also enzymatic breakdown of these molecules. You can see all the places where you have arrows. Here are essentially where the molecule is constantly attacked. This is also happening in living cells, but here we have all kinds of repair mechanisms that are keeping the molecules intact. So this means that when we work with ancient DNA, instead of having nice long DNA strands that are easy to sequence, easy to analyze, and easy to extract a lot of information from, well, we have a pool of very degraded molecules, and it's more challenging, it's much bigger puzzle to try and piece this together in order to get our information. In terms of working with this, I mean, it's a big problem, of course, when you have highly degraded molecules to do PCR, because you cannot, see if this works, you cannot place your primers on very short molecules. There's simply not room for placing your primers and having your insert, which implies that very often the standard PCR-based technology, where you amplify single genes or certain sections of the genome, will fail with the ancient DNA. So it's a big problem for PCR. But in terms of next generation sequencing, which of course we've switched to over broad scale these years, it's not really a problem, because you can sequence very short fragments with the next generation sequencing. You are getting millions and billions of DNA sequences very, very rapidly, and you can essentially sequence, as I mentioned, very, very short fragments. So the problem here more becomes a bioinformatical problem. You can see various examples of these fragment length distributions that could, the blue all the way to the left is a highly degraded sample. And then you have various states of DNA preservation in these samples. And here the issue becomes bioinformatical, because as soon as we get below roughly 30 base pairs, we don't have enough information anymore to be actually able to place the sequence, assign it to a certain species, or place it at a genome, at a position in the genome, which means that it's molecularly we cannot use the sequences. So even though we can produce the data, we cannot actually analyze it and use it for anything. Another thing that concerns us a lot in our work is we talk about the endogenous DNA content, which is essentially the proportion of DNA in our sample, the proportion of DNA that derive from the target species. So if it's a human bone we're working with, it's the proportion of DNA sequences that we can determine as being of human origin. This is a big problem in a big issue in ancient DNA research, because when we get, as for example, a skeletal element, a tooth or a bone that have been lying in the ground for thousands of years, these bones have been colonized by microbes, bacteria, fungi, which of course, they live and die inside the bones, which means that they also deposit the DNA inside the bones. And next generation sequencing, by default, cannot really discriminate between these various sources of DNA. We just sequence any DNA that might be available to us in the sample. And this implies, as you can see here, that when we have a modern DNA sample, we will normally have a very high proportion of this endogenous human DNA in the case of humans. Very easy to get a whole genome sequenced very rapidly and quite cheap. But when we're dealing with an ancient DNA sample, ancient tooth or bone, with this human or this target species DNA proportion, the endogenous proportion is often very, very small, often less than 1%. Then we have a lot of microbial DNA, and we have a large fraction of DNA that we cannot necessarily determine. So this, of course, means that ancient DNA work, at least if we want to work at a genome scale, is very expensive, because every time you sequence 100 sequences, you only actually get one you can use, and you can throw away the rest in many cases. So that's another issue that concerns us a lot. This is not really a problem for PCR again, because PCR is very good at if there are a few molecules present, target molecules, well, the primers will bind to those in your extract. But it is a big problem for next generation sequencing, where you sequence indiscriminately across the genome, at least when you use it in the sort of its default version. So this means that the big population studies we're doing these days, they can really be tough, because you need to screen through a lot of samples to actually find the few good ones. This is just an example from this Bronze Age paper that we published a few years ago, where we had to screen through the preservation state of more than 600 samples to actually try and find the few samples, the few percentage of samples, or the few samples where you have, say, more than 5% human DNA in them, because these are the ones we can actually use for a genome scale analysis downstream. So it's a very tedious, painstaking process to shotgun sequence through a lot of samples at low depth to try and find the good ones. Then you select the good ones, and then you sequence them at higher depth and get the genome data downstream. So you actually have some analysis to perform. So this also implies that there are some key steps to a successful ancient DNA project. First of all, you have to know and target the best type of skeletal element. It's very important. It matters a lot which element you're actually going for. I will show this in a little while. You have to target the best preserved samples. You need to be able to identify which of these samples has a preservation state that warrants molecular preservation. And you have to then use the best sampling practice, the best sampling method, because this is also extremely important. It matters a lot which method you actually use and how you do this. And then you can say there's a lab work, next generation sequencing, bioinformatics. You come up with a hypothesis to test, and you publish in Nature. That's sort of the way it goes often in ancient DNA research. It sounds a little bit arrogant, but for some reason the high impact journals really like ancient DNA research. And of course, the thing about coming up with a hypothesis to test, this might seem a bit late in the process of proper science to do that, and sometimes we also have it up front, I have to say, which is the way it should be. But you have to remember that we work with a lot of samples here that often don't work. So we can very often in these projects come up with a lot of interesting hypotheses to test, and at the end of the day we have to throw them away because the material doesn't work. And in the end you have 3 samples from Siberia that work, and these are the ones you end up building a project around. So this is said halfway as a joke, but there's certainly some truth to this. Anyway, I'll focus here mostly on the 3 first steps here. So a few words about substrate and sampling method. We use teeth a lot in our research, and for quite a few years when I sampled a tooth, I would cut it open like here, and then I would specifically target the dentine layer inside the tooth root. You can see that on the cross section to the right. And because we thought, okay, this is the dentine inside the tooth, it's very protected from contamination from the outside, for example, so it's probably the best thing to do. So we drilled it out with these small drill bits and extracted DNA from that. It turned out to be a very stupid thing to do, because at some point we realized very, very late in the process that actually by targeting the outer layer of the tooth root, the cementum layer, it really increased this endogenous DNA content, in this case the human DNA content, quite a bit. This figure here is just showing that. On the y-axis you simply have fold enrichment, so how many times the sample is getting better when you're sampling the cementum compared to the dentine layer. And you can see here for many of these samples, there are 14 samples here, you get as much as more than 5 times more endogenous human DNA content by targeting the cementum layer in contrast to the dentine layer. So there's a lot of money to be saved here when doing it like that. And this, have we known this from the start, it could really have saved us a lot of time and money. And it's a nice example of how you can have the best labs in the world, you can have a very expensive sequencing machine and the best bioinformatics to work on it, but if you don't know to drill a bloody tooth in the lab in the first case, then you don't have a project. And yeah, low practical stuff. Another thing we realized in the same study essentially is that we introduced a pre-digestion step, which is something very intuitive and easy to do. Essentially, you just give your sample a very brief enzymatic treatment to essentially digest away all the dirt in the surface of the sample. So we use proteinase K in this case to digest away all the dirt and the microbial DNA that might be sitting on these surfaces. And of course, this by doing so, it's essentially a way of cleaning up the sample. Let me see if it still works. Okay, I cannot talk about, so this was just an example of how targeting the teeth and targeting the right way is really had made a big difference. But I cannot talk about ancient DNA sample without mentioning the petrous bone, because this is really, I would say, the new black in ancient DNA research these years. It's a bone sitting inside the human skull, protecting the inner ear, and it's extremely dense. And because it's so dense, it also preserves DNA extremely well, and it prevents microbes from actually coming in and you can say polluting the sample with environmental DNA. And there are several papers demonstrating how this bone is extremely well suited for ancient genomic research. We just published a paper, I think it was yesterday or the day before that, showing that it does have a trade-off targeting this petrous bone, because it's so dense, you cannot actually get any ancient pathogen DNA from this bone. It only preserves the human DNA essentially. So if you go for the teeth or the postcranial bones, you can actually find traces of ancient pathogens that these humans have been carrying with them, plague, for example, or tuberculosis. So also understanding and studying evolution of past diseases. So I mentioned preservation state. It's not actually that difficult to determine if a sample is well preserved or poorly preserved, and it really makes a lot of difference when you go for this, or when you try and determine this before the sample. This is an example of poorly preserved teeth. I would say they're kind of chalky. You don't have much collagen left in these. You can even break them with your fingers if you try to do that. And you can see here that when you take these poorly preserved teeth and sample them from individual skeletons, and also take petrous bones from the same individuals, you will have a lot more DNA inside the petrous bones than you will actually have in the teeth. That is shown on this graph. So the clustering of these bars are individual skeletons, and the green bars are the teeth, very little DNA amount, and the orange here are the petrous bones. However, if you have skeletons with well-preserved teeth, like they're seen here, lots of collagen, has this nice yellowish color to the roots, and you can almost see a little bit of light shining through somehow. Here it doesn't matter that much. The petrous bone might still be better in some cases, but the teeth perform really, really well. So it just means that when you start up an ancient DNA project and start the sampling, it's very important to take a good look at your sample to try and assess which way you should actually do this. Another way of looking at this, I had some of our lab technicians writing down, whenever they did the sampling, just very crude measures of how they saw this sample, if it was well preserved, if it was poorly preserved, and this happened before we sequenced them. And I divided this into 3 crude categories. Well preservation, that's when they had noted words like hard, well preserved, perfect, solid, words like that. Okay, then I pulled them in the well preserved category. Intermediate, that's when they had like semi-soft, rather solid, okay, but not perfect. And then poor preservation were assigned with words like soft, mineralized, chalky, no cementum layer visible, or the tooth crushed between the fingers or whatever. So very crude measures of just looking at this. When you look at the endogenous content of these samples and try and relate that to these preservation comments, you see quite a nice relationship here. So the well-preserved samples, the good one, actually turned out they had almost 15% of human DNA in them. This is quite high in ancient DNA context. We're very happy with samples that have this high amount of endogenous DNA. The intermediate, a bit lower, we're down to an average of 8% here. And when you go into the poor preservation samples, well, we are below 5%. And when you break these numbers down even further, you compare the well-preserved state 1 with the poorly preserved state 3, you can see there really is a very big difference in either end of the spectrum. In the state 1, we have a lot of samples that are up to 50% endogenous human DNA. Very happy with such samples. We can do an ancient genome in no time. Whereas in the poorly preserved samples, we don't have very many of these samples. And on the other end, we have a lot of samples, more than 30% that are below 0.1%. These samples cannot really be used to anything from our perspective. So the whole idea, and what I was told when I started working with ancient DNA, is that you cannot predict ancient DNA preservation. You just have to sample and try. It's a myth. It really is. I mean, you need to look at the sample before you start the work, and you have a very good idea of this sample will actually work or not. And there's no idea in going out and do destructive sampling of hundreds of ancient skeletons and essentially destroy them for exhibition and reduce the exhibitional value if they look poorly preserved. Sometimes, okay, you can be lucky that they actually work, and if it might be a very spectacular sample, so you want to try it anyway. But certainly you have some numbers here now to assess the chance of it actually working. Okay, a few words on DNA preservation and how long it is preserved, and I think this is probably why I was invited to talk here today, because in 2012 we had some data to look at the DNA decay, essentially, the rate of DNA decay based on ancient fossils or subfossils. In this case, it was during my PhD time, I worked on this big extinct bird from New Zealand called the moa bird, weighing over 250 kilos. We had a lot of bones from the same swamps, essentially, where they had been preserved for more than 600 years, up to around 5,000 years. And I realized it was the extinction process I was interested in and the evolutionary relationship to other big rat-tailed birds and so forth. But I realized that during all this data we produced, we actually had a very good setup to try and address DNA decay. Because we did quantitative PCR on these samples, implying that we knew roughly how much DNA, how many copy numbers DNA of that particular gene we were targeting was present in each sample. And because we also radiocarbon dated all these samples, we had we could try and correlate, of course, DNA amount with age of the specimens. And as you see here, I mean, it's not the best correlation of the world, but it's certainly there. We have an R-square value of around 40%, right? And notice this is a logarithmic scale, which means that we have an exponential relationship here. It's not a huge surprise, this. This is what people have been hypothesizing for a long time, that DNA follows an exponential decay curve. But this is the first time it was actually shown on ancient data. I mean, real long-term DNA decay could be shown here with this data. A lot of noise. It's a biological system, but still the overall relationship is quite clear. And the reason why this has been tough is because this is a very rare setup to have, that you have hundreds of samples from the same area exposed to the same environmental factors and all of them radiocarbon dated. Normally in ancient DNA projects, you will have a few bones from one cave, a few bones from another, and then you're really comparing apples and oranges. But in this case, we could address this. When you have an exponential relationship, you can come up with some sort of equation. We introduced temperature into this because we knew the temperature these have been buried. We could calculate that. And based on that, we could start calculating DNA decay rates at various temperatures. This is quite hypothetical, I should say, because these things, environmental factors, can change over time, and there are other factors at play here, like pH and everything. But nonetheless, this is the best we had at hand at that stage, and probably the best estimate still around until something comes up with something better. Anyway, what we could see here, that by looking at these decay rates, is that we could calculate the half-life. And one important aspect here is, of course, the colder it gets, the slower the half-life is. I mean, DNA decays much slower at lower degrees. But one important observation is here that even if we say permanently under -5 degrees Celsius, it doesn't take more than 6.8 million years in this case before the DNA is completely disintegrated. I mean, down to fragmented down to 1 base pairs. 6.8 million years. I mean, it's an estimate, but it's the best we have so far. And please remember that I said that when the DNA is below 30 base pairs, we cannot use it for anything analytically. I mean, that's where the limitation is, and that happens much before 6.8 million years, I should say. So I would estimate based on this, we should probably be able to push the 1 million year boundary if it's preserved really, really cold, but we will not ever be able to find dinosaur DNA. I would love to be proven wrong on this, but so far we haven't seen any indications on this. And this means that a lot of these studies that came out in the 90s, I don't know if you remember these, I've seen them, where people were looking at 120 million year old beetle DNA, 15 million year old DNA from a plant here, and here you have DNA from the dinosaurs, right? The Scott Woodward and colleagues published this in the 90s. This is the paper that made Michael Crichton write his novel and Steven Spielberg make the Jurassic Park film. I mean, based on my own results and what other people have also been looking in, I think this is your dinosaur you have right here. I mean, it is our best bet is that this must be contamination one way or the other in these samples here. This, I would say, is the current reliable world record. The genome that was published a few years back now of a 700,000, roughly between I think they say between 500 and 800,000. It's difficult dating that old material, but roughly 700,000 year old horse that had its whole genome published. And that really tells, and not surprisingly, this was found in northern Alaska, where it has been buried in cold for this entire period. And that is one very, very important notion, is that the colder it is, the better the DNA is preserved. We see that all the time in our work, that when we get samples from Siberia, Alaska, Greenland, there's a good chance of it working. Whether we get it from southern Europe, Africa, or the tropics, then it's much more challenging to actually get something out of it. And of course, this is also important for preservation or biobank perspective, is and that is why we keep our ancient DNA sample in big walk-in freezers at our institute. Okay, do I have a couple of more minutes? Okay. I'll keep going. Looking at temperatures, and this half-life paper introduces another concept, that you can actually take these fragment length distribution here and look at DNA decay based on those. So the steeper this exponential curve is, the more degraded your sample is. You can see how it degrades the longer the DNA is, and this is essentially because every time you lose one long DNA molecules, you get 2 shorter ones. That's a simple way of saying it. So you get this exponential accumulation of shorter DNA fragments. But the steeper this is, the more the higher the more the DNA is degraded. And of course, you can then calculate these decay rates. When you know the age of the sample, you have an actual rate. And you can look at samples from different preservation environments, and this is just looking at very crude way of looking at samples from different. You can see different temperatures around the world, and you will see here that the molecular half-life is very closely correlated to how warm it is. I mean, when you sample from the Caribbean, you have a half-life of only 170 years, 400 base pair fragments. So DNA decay is really quite rapid in this case. And this is another interesting aspect, because we meet a lot of people, archaeologists, who talk about in situ preservation. They'll keep the bones in the ground so we know we can always excavate them later, come back to this area, and it can make a lot of sense for many purposes, in particular if you don't have money to excavate, right, or funding. But in this case, I mean, if you want to use these samples for anything that related to molecular biology, it's much better to actually pull them out, or at least take a subsample and put them out and chuck it in the freezer and then revisit the sample later. Because if you leave this in the ground another 100, 200 years, 300 years maybe, then there's no DNA left to sample. Finally, I think I had in the abstract that I also work a little bit on ethanol and formalin preserved material. I'm very interested in reptiles for some weird reason. so I try to, at the Natural History Museum, we have a large herpetological collection. And I've recently had a dabble with this, and of course, coming from an ancient DNA background, I very carefully try and assess molecular preservation in this material before I actually start any genomic work. And I'm just showing you a small case study here, a few results from working on an extinct population of the Aesculapian snake that is nowadays is distributed in mid and southern Europe, but have some extinct populations in Central Europe and also in Denmark. And we have 3 specimens at the museum from this extinct population that went extinct roughly 200 years ago. And they've been lying there in ethanol for 200 years. And I tried to sample these for DNA using a very carefully optimized ancient DNA extraction method. These specimens very often fail with PCR-based methods, which is why I thought, okay, let's, instead of just saying that we can never get it to work, let's try an ancient DNA method on these. And again, a lot of numbers here, but you can see the total number you have here is the total number of DNA sequences that we've generated just in this screening run from each of these individuals, and then we can assess various measures of DNA quality here. In particular, important here is the C/T damage rate, which is it's a way of assessing one of the damage parameters that are very relevant to ancient DNA research. It looks like mutations in the sequences, but essentially it's just a deamination of the cytosine, which makes it look like a thymine when it's been through a PCR reaction. Anyway, long story, and you can see here these samples. What I was very surprised to see here, that these samples, only 200 years old, 150 years old, was just as damaged as some when I work with a 40,000-year-old Neanderthal or something. I mean, it was really amazing to see the level of DNA degradation in these samples. So clearly, there's something very suboptimal going on in this way we preserve this material. You can look at the fragment distribution here. There are a few bioinformatical biases. These are the bumps you see, but nonetheless, you can clearly observe the overall exponential correlation. And I don't know if you can see the small numbers, but the average fragment length here is something like 40 to 50 base pairs on material that is only 150 or 200 years old. And I was very surprised to actually see this. Extremely damaged. This could have been ethanol, which is actually not very good for long-term storage. It could also be formalin exposure. We don't really know. A lot of this material has been formalin exposed to formalin in some way, either when killing the animals or when preserving. We don't know for these particular animals, but it's very likely. And formalin is very good for protein preservation, but for DNA preservation, it's very, very bad. Okay, this I'm going to skip, and I'm just going to summarize. Last slide. So, picking the best substrate for ancient DNA work, petrous bones and tooth cementum are both excellent substrates, and these are the ones, this is our best practice now. This is the one we really try to target. Sample petrous bones when tooth preservation is bad. There's no point in sampling postcranial bones or teeth when the preservation is bad. Then you should always go for this one if you want something that works. So you can sample either when tooth preservation is good. Few words on preservation. If a skeletal sample looks poor, then it is poor. I mean, it's well worth knowing, and you'll save a lot of people a lot of time when they address your collections or when you want to work on this material. If you keep this knowledge at hand, long-term DNA decay can be modeled as a rate and is highly temperature dependent. All ethanol preserved specimens are highly DNA damaged, at least the ones I've looked at, and may require carefully optimized ancient DNA methods and next generation sequencing to work. Thank you very much.